The overall goal of this experiment is to investigate the sensitization to various grass pollen allergen components in southern China.
This method can help answer key questions in the allergy field about how individuals become sensitive to grass pollens.
The main advantage of this technique is that the use of recombinant or purified allergen molecules can circumvent the occurrence of confounding reactions, providing a better test accuracy.
The implication of this technique is then toward the diagnosis of allergic rhinitis and asthma, as many of these patients are sensitized to common detectable allergens.
Though this method can provide insight into allergic airway disease, it can also be applied to other types of allergies, such as food allergies.
We first have the idea for this method when we find that very limited data on CRD exists for Chinese Southerners.
Demonstrating the procedure will be Nili, a technician from my lab.
After retrieving the serum samples of eligible patients from the biobank, turn on the fully automated immunoassay analyzer and the built-in information data management computer. With the primary power on, switch the instrument to the system power and wait 3 minutes for the built-in software to start. Then add 140 microliters of serum to 1 vial per allergen and/or allergen component test and label each vial of serum with an